Welcome to the latest Novogene webinar, introducing NovoMagic, Novogene's online RNA-seq bioinformatic analysis tool. Today's webinar is different to our usual beginner's guide. It will focus on Novogene's online RNA-seq bioinformatics analysis tool, NovoMagic, discuss how to navigate NovoMagic's functionalities and provide tips and tricks for its successful applications. A bit of housekeeping before we start. The presentation will run for around 40 minutes with 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. If we don't have time to answer your question, we'll come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We will share the slide deck and the recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display the certificate. You can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this as help us to plan future webinars topics. I would now to introduce your host for the webinar, our scientific content specialist, Eva. Eva, over to you.
Thank you, Miranda. Hello, everybody, and thank you for joining our webinar today, introducing NovoMagic, Novogene's new online RNA sequencing data analysis tool. My name is Aoife. I'm Novogene's scientific content specialist and your host for today's webinar. Shortly, I will pass you over to Yue, who will talk you through the applications and functionalities of NovoMagic. I appreciate many of you here are probably customers of ours already looking to learn how to use the NovoMagic tool. But before I hand over to Yue, I'm going to speak a little bit about Novogene, just in case there's anybody here joining us for the first time. So Novogene was founded in 2011 and has been growing rapidly ever since. Our company mission is to advance genomics and improve life. And as you can see here, our services cover a huge range of applications with real life implications, from complex disease research and oncology to food and water safety. Our European Sequencing Centre is based in the Cambridge Science Park in the UK. Here we have our state-of-the-art lab equipped with the latest Illumina NovaSeq 6000 sequencing machines. And we have always provided fast turnaround times and high quality data, but that has improved even further with the recent installation of Falcon 2, which is our new NGS intelligent delivery platform. This has increased our monthly capacity by 200% compared to manual steps. We also have our supercomputing cluster, so we can offer bioinformatics services and store your data in the UK. However, at our heart, we are a global company. We can offer services across Asia and the US and of course Europe. Falcon 2 has now been deployed across a number of our labs, standardizing our services globally. With almost 2,000 employees and serving over 5,600 customers in world-class universities, biotech companies, and hospitals, we can provide sequencing for the smallest or largest of projects. We provide a full range of next-generation sequencing services, such as whole genome sequencing, whole exome sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. These can be applied to a wide range of research topics, from cancer and rare disease, microbial cell factories and gut health, to agriculture and animal health. The focus of today's webinar is on NovoMagic, our new online mRNA data analysis tool, which is applicable to human, plant, and animal mRNA sequencing data. mRNA sequencing provides data on transcript expression levels, differentially expressed genes, and allows for the study of these elements under varying environmental and disease conditions. The data produced can provide information on disease mechanisms and biochemical function of expressed genes, and NovoMagic enables customization and reanalysis of this data. Many other companies offer these services too, so why Novogene? There are a lot of good reasons why you should trust Novogene with your project. As I mentioned, we've had a really exciting development recently in the installation of the Novogene Falcon 2. This has now been deployed across our Chinese, US, and Cambridge labs, standardizing our global service. This new technology was developed in-house by Novogene and has already massively increased the capacity of the Cambridge Sequencing Centre, And it will also increase the quality of the data that we can produce because it is entirely automated and will remove the possibilities of any human error. With this new addition, we're now able to offer our fastest ever turnaround time for RNA and whole genome sequencing, just 10 working days from sample arrival to data release. Falcon 2 really highlights the innovation that we strive for here at Novogene in an effort to keep pushing the frontiers of genomic research. NovoMagic is another example of this innovation and an example of how we endeavour to provide solutions based on our customers' needs and to continuously introduce new and helpful tools to the genomic industry. The benefits of our in-house innovation are clear in our record in the scientific community, which we're very proud of. We've been cited in over 790 research papers with a total impact factor of over 6,120. And we're also driving the NGS industry forward with over 37 NGS-related patents. Our technology is helping to make the NGS process more efficient and effective, all the while improving the quality of the data that we deliver. The Novogene community keeps on growing. Our expert sales team speaks over 10 languages and is located across Europe, ready to assist you with your next project. If you're not ready to send us samples yet, you can still benefit from our educational outreach to learn how NGS can work for your project. This includes webinars such as this one, and we also recently wrapped up our Southern European Roadshow seminar. But we are planning on picking it back up in the new year, so keep an eye out on our social media for future seminar announcements, and hopefully we'll be visiting somewhere near you soon. And if you would like us to organise a seminar at your institute, please do get in touch with us. I would like to quickly introduce our European sales team. So as I mentioned, they are based across the continent and ready and waiting to help you with your next sequencing project. If you would like to speak to your local representative, you can email us at info@novogene-europe.com and you'll hear back from them. With that, I would like to thank you for your attention and I'll hand over to Yue. Yue is one of our product marketing managers at Novogene and is an expert in all things bioinformatics. So with that expertise, she's going to guide you through the applications and functionalities of NovoMagic. Over to you, Yue.
Thank you, Aoife, for the nice introduction. Please just bear with me and let me just share the screen. Okay. Hello, everyone. This is Yue from Novogene Europe. And today I'm glad to introduce you with NovoMagic, which is Novogene's online RNA-seq bioinformatic analysis tool. So before we start the talk, I'll let you know that today's talk is not about coding. It's more related to the introduction of this online platform and how to use it. So NovoMagic is an online RNA-seq BI tool for the Novogene client. I know this is very different from the previous beginner's guide educative webinars that we run, but I will also use the standard analysis of RNA-seq as example, guiding you through the functionality that NovoMagic covers. Okay, so what is NovoMagic? NovoMagic is an after-sales analysis cloud platform independently developed by Novogene. So currently, it can help our client to reanalyze and customize the Novogene RNA-seq analysis results totally for free. And all analysis on NovoMagic allows customizable parameters and the comprehensive toolkits, including quantification of gene expression, identification of DGEs, functional analysis, and a lot more. So far, we have run over 40K projects on the platform from different departments, and the functions are now limited within the range of RNA-seq, but we are continuously updating the platform to widen the scope of the analysis types. So here I'm also showing you the sidebar of NovoMagic, which includes the functionality of it. And there are a series of reasons that NovoMagic might be beneficial for you. So it could save the time of building up your bioinformatic analysis environment and also reduce the cost of communication with your bioinformatician or service provider, even when you want to just simply adjust the parameters. And as mentioned, you can get unlimited access to the platform and your project data and all future updates of the platform. Here I'm also showing how NovoMagic is integrated into Novogene's customer service system. Also, it's quite convenient to use the built-in FAQ and tutorial. So the two of the highlights of the platforms are the intuitive and user-friendly interface and the ready-to-publish data that can be generated from the platform. And here's how the interface looks like, but I'm also going to show you a short demo at the end. I understand that you still have some questions after this short intro. Let me just use eukaryotic RNA-seq analysis as example to go through the workflow from the quality control to functional analysis, which mostly align with the functionality of NovoMagic. And in the end, we will come back to the platform and show you a demo about how to use it. So before we jump right into the analysis part, let's review the concept of RNA-seq. So RNA-seq is utilizing NGS to review the presence and quantity of RNA in a biological sample at a given time. And the applications depends on the biological questions you want to answer. So for characterization, the common applications could be genome annotation. For expression, this could be gene expression and differential expression, etc. On this side, we're also showing what those information can be applied for. For example, for biomarker discovery or host and pathogen interaction, and a lot more. More specifically, at Novogene, we have different solutions for RNA-seq. Here, we're just listing part of them that are more frequently used for the gene expression analysis. So we have RNA-seq for the bulk RNA-seq analysis, and we have link RNA-seq, circular RNA-seq, and a small RNA-seq. So if you want to study all of these four, it would be a total RNA-seq. And if you're struggling to get enough material, if you have a limited starting material for RNA or cells, you could also use the ultra-low input RNA-seq that we offer. If you're trying to find a solution for single cell, we also provide the primer library sequencing for you. But today, we're more specifically cover the analysis of mRNA-seq. If you're interested in any other kinds of RNA-seq analysis, please visit our website for other series of beginner's guide. And more precisely, there are a list of products that are applicable for NovoMagic, including the eukaryotic mRNA-seq for human, mouse, and other eukaryotic species with reference genome available. And it's also okay if you have just a standalone RNA-seq BI analysis with that. That's also okay for using NovoMagic. So let's take a closer look at RNA-seqBioinformatics analysis workflow. So there are 5 major steps, from the quality control to mapping to expression analysis, functional analysis, and characterization analysis. So there are star marked for the functions that covered at NovoMagic. Okay. Then let's go through them from the beginning, which is the quality control. So first thing first, how does the raw data look like? And as you can see, each read contains 4 rows, and the first row, which contains the sequence ID and also the library index. Whereas for the second row is the read basis with the separator, and the fourth row will be the quality scores. So the reads are generally stored in FASTQ files, and for pair and reads, there's a second file with a 1-to-1 corresponding header and reads. So each file can contain millions of reads, and they are mostly always compressed to save space. So it will be in fastq.gz extension. And here I'm also showing a relationship between the sequencing quality and the base call accuracy. So a quality score of Q20 represents an error rate of 1 in 100. meaning every 100 BP sequencing, the read may contain an error with a corresponding call accuracy of 99%. And there are just 3 major aspects we look at the quality control, which includes the error rate, the base content, and the data filtering. The software we use for QC is called FASP. So for error rate, we're looking at the position-based quality, as we all know that errors tend to accumulate during the sequencing. And this is the normal look of error rate. For GC content, we're looking at different things, such as no separation of GC, no split at the end, and no vibration. And as we know that a large variation of sequencing error in the first 6 to 7 bases is allowed, considering the usage of random primer in the library preparation. Okay. Whereas for data filtering, we would filter out the rays contaminating adapters and ambiguous read, also low quality reads, and then we will get the clean reads out of it. So after the QC, the clean reads will be ready for the next step, which is the read alignment mapping. And for the mapping part, essentially we're trying to determine the location from which each read originated. And with studying an organism with a reference genome, it is possible to infer which transcripts are expressed by mapping the reads to the reference genome or transcriptome. And here's the case of different types of RNA-seq reads, and the red reads are mapped entirely within an exon, and the blue reads spanning over 2 exons, whereas for the purple reads spanning over more than 2 exons. And the principle of the spliced mappers would be first to identify the reads spanning a single exon, and then to identify the spacing junctions on the unmapped reads. And the software we use for the mapping step is called HiSet2, which uses a graph-based approach to index the reference genome and combine with the bowtie tool algorithm for alignment, and provides the alignments known for its accuracy and speed and sensitivity. So the input for the mapping part is the clean reads that we get after QC, and we also need to provide the reference genome, which is in FASTA file format. And the output will be a binary version of a SAM file, which is called BAM files. Since the output of BAM file is not readable, it is a binary version with just showing the summary of the mapping result. As you can see, for each sample, we have the situation, the summary of the total reads number and the uniquely mapped reads, multiple mapped reads, and also the total mapping read, uniquely mapping reads, and multiple mapping read. And this is what we get after mapping. So after the completion of mapping, the analysis moves forward to the expression analysis, also called as quantitative analysis. And from this part, it is already available on NovoMagic. So all of these functions, you can find it on NovoMagic. Speaking of gene expression quantification, the simplest approach to quantify the gene expression by RNA-seq is to count the number of reads that map to each gene using programs such as feature counts. So for feature counts, it counts reads that map to a single location, which is what we just mentioned, uniquely mapping, and assigns the reads to a gene or exon. So the input of the feature count is the BAM files we get after mapping, and the GTF file, which is the annotation file. And the main output of feature count is a table with the counts. For instance, the number of reads or fragments in the case of pair end reads mapped to each gene and in the provided annotation. So after getting the read counts of each gene or exon of all samples, there's one critical step before the differential expression gene analysis, which is called normalization. But to better explain the necessity of normalization, we will first need to review the idea of coverage in NGS. So for NGS coverage, it describes the average number of reads that align to or cover known reference spaces. So we have two types of coverage common here. One is the depth of coverage. It is about how strong is a reference covered by the sequence fragment. It is often expressed as 1, 2, or 3 times coverage. And here it's about how to calculate it. And on the other hand, we also have the breadth of coverage. It is about how much of a reference is covered by the short reads. So the breadth of coverage is the percentage of bases of a reference genome that are covered with a certain depth. So for example, we could say 90% of a genome is covered at 1 time steps, and still 60% is covered at 5 time steps. And the more commonly used coverage is the depth of coverage. Okay. So what exactly is normalization and why does it matter? So for instance, we have example here. So we have the treated sample and we have 15 million total reads with a depth at 5 times. And the control samples, we sequence 10 million total reads with a depth at 2 times. So would you think the transcript of the treated sample express more than the control sample just judging from the depth? But actually, if you also take the total reads into consideration after calculation, the treated sample has just half the transcript expression of the control. So this is about the comparisons between sample, also the importance of taking into consideration about the total reads number sequence. But what about different genes within one sample? And the read counts also depends on the total gene or transcript length. So consider that we have 3 different genes with different lengths, gene A, B, C. They have different lengths at 2, 1, and 3 kb. And we need to first standardize the read counts using the transcript length and get the comparable read counts for each gene. And therefore, we can get a straightforward read counts result from comparison. And after this, all of this, you might be wondering, what are the concept and the name of the normalized read counts? So in fact, there are different expression value here. After the normalization, what we have is, they're called RPKM, FPKM, and TPM. And RPKM is short for reads per kilobases of exon per million of map reads. It is more for single end read normalization. Whereas for FPKM is short for fragments per kilobase of exon per million of map reads is for pair end read normalization. It is also used in NovoMagic for normalization using DSeq2. Whereas for TPM is short for transcripts per kilobase million. And it's actually very similar to RPKM and FPKM, except the order of the operation. It can be used either for single or pair end reads. So the operation order is actually normalized for gene length first, and then for the sequencing depth. Okay. So I know that you might be familiar with one or all of them, but if you just want to, let's say, use TPM for your analysis, we do have a toolkit, a function in NovoMagic allows for the conversion from read counts to FPKM or TPM. And then after getting the expression value of different transcripts, we could move further to the correlation analysis. And there are actually different ways of determining the intra and the intergroup sample variability and outliers. So a global view of the data allows for the characterization of variation between the replicates and whether the investigator-defined experimental groups show actual differences between the groups. So both PCA and sample correlation analysis can help to identify outliers that were not excluded during the upstream steps, such as alignment. So these are useful for realizing the overall effect of experimental covariates and batch effect. So for example, we could see from the Pearson correlation between samples. When the R square is closer to 1, the samples are more correlated. And this graph actually shows a good reproducibility because it replicates I correlation. Whereas for PCA analysis, you can see the samples from the same group, they are grouped nicely on the graph, also showing the good reproducibility. Okay, so after the correlation analysis, let's come back to it and remember that we touched on the point of concept of normalization, and now we move towards the differentially expressed genes analysis, which are the highlight of RNA-seq. So the software we use is called DE-seq2, and the input of DE-seq2 is a table of raw recons, and the output will be the results table that I'm going to show you later. Okay, so I'm not going into details about how the software works, only the principle of it. So first, it will calculate the group mean, which is the pink circle for the different groups. Also calculate the global mean, which is the yellow circle, and calculate the deviation between the global mean and the group mean. So if it is more separated like this, it means it has significant difference. If it is closer like this, it means that there's no significant difference. So DE-seq2, which is perform like this, we first perform an internal normalization. And to calculate this means that we just mentioned, and the count for each gene in each sample is then divided by this mean to correct the library size and the RNA composition bias. And then it will use the hypothesis testing for the significance testing. And finally, making the adjustment of the p-values. Okay. So the results table of DE-seq2 looks like this, and each row contains the information for each gene or transcripts, and with the first column contains the gene or transcript ID. The other columns are the statistical value for the gene or transcript. And after the annotation of those DEGs, the gene list will be ready for visualization and further analysis. And here we're also showing an alternative option on NovoMagic for the DEG analysis, which is EdgeR. And here's the interface where you can perform DEG analysis on NovoMagic. Okay. So here we're listing different ways of visualizing the DEGs that we just got. And one of the popular one is called the volcano plot. And volcano plot is often the first visualization of the data once the statistical tests are completed. And this plot shows data for all genes, and we highlight those genes that are considered DEG by using threshold for both the PHS value and the fold chain. But if you have more than 2 samples or groups, gene clustering is used to classify those DEGs with similar expression patterns for the subsequent analysis of data from experiments, such as time courses or multi-group comparisons. Also, the Venn diagram is a straightforward way of showing the co-regulated genes. And here we just show an example of the experiment setup. So after having a list of the DEG genes, we can just assign function to it, and this is what we do as enrichment analysis. And this can be used to facilitate the biological interpretation of DEG analysis results. As you know, these are also available on NovoMagic platform, and here I'm just listing the sidebar where you can find geo, CAG, reactome, and a lot more. So, yeah, so here we're just telling you more about how to do the enrichment analysis, and the software we use is Cluster Profiler. And what Cluster Profiler does is to determining whether there's enrichment of non-biological functions, interactions, or pathways. and identify genes involvement in novel pathways or networks by grouping genes together based on similar traits. Okay, so use global changes in gene expression by visualizing all genes being significantly up or downregulated in the contents of external interaction data. So the databases we use for the functional analysis, for the enrichment analysis, actually there are a lot, but we're just listing two of them, which are GEO and CAC. So GEO and CAC are the most frequently used for the functional analysis, and they're typically the first choice because of their long-standing correlation and the availability for a wide range of species. And please note that there are also other ontologies and pathways databases available, such as DisGenet, Reactome Pathway, etc. And here is the graph that you can get after the enrichment analysis. For GEO, we're showing a GEO enrichment scatter plot, and for the CAC, we're showing a pathway graph. So for the GEO enrichment scatter plot, the y-axis is actually the GEO terms, and the side of the dot is the number of the DEGs, and the color of the dot is actually the range of the PHS value. Whereas for the CAC pathway, you can see the CAC genes sits in the different pathways. It might be a bit small, and the colored boxes actually the genes in the pathway that are differentially expressed. So that's what we get from the functional analysis. And here's just a short summary of the standard analysis that we have for messenger RNA-seq. And from the quality control to mapping, and as I just mentioned, NovoMagic will take it from here, from the expression analysis and functional analysis. And you might be wondering that why characterization is not included. And the reason is that because for characterization analysis, it is more intensive in terms of the computing capacity and also the resources. And the input file for those characterization analysis is actually the BAM file, which is a much larger file comparing with the input file for the expression analysis, which is such as the recons file. So it's taking more resources. So far it's not included on NovoMagic platform, but it's definitely included in our standard analysis. Okay. And finally, we are approaching the point of talking more about NovoMagic. I have already talked about this at the beginning, but just for those who join a bit later, just a short introduction. So NovoMagic is an after-sales analysis cloud platform developed by Novogene, and it can help our client to reanalyze and customize the Novogene analysis results totally for free. And here are some of the advantages of the platform, including the user-friendly interface and ready to publish data. It's also free of charge for our clients. You also get unlimited access and it's convenient to use. Okay. And here's a bit more detailed. So let's just divide this into two parts. So let's talk about the functionality. Sorry about typo. And here you can see this is just a lot of analysis point, such as for the clustering, the correlation analysis, also to produce the volcano plot, heat map, violin plot, and a lot more. Also, you get to do a little bit changes for the tables, no matter what format it is and what content it is. Okay. Whereas we also have the guest mode, and it contains the three popular functions, which are the recount to FPKM or TPM, and also volcano plot and heat map. So it means that if you're not available to the full functionality, you can also try the guest mode. But more detailed, let's have a look who can use the full functionality. So for Novogene clients who have CSS accounts and have RNA-seq projects with samples or data arrived at Novogene after the 20th of June this year, And the NovoMagic platform functions for those projects after whose BI analysis are finished. Whereas for the guest mode is easier, it's just for Novogene customer who have already activated the customer service system. So if you have any doubts considering this part about if you can use it, please do not hesitate to ask questions and we will just be very happy to help you. Okay. And here I will be showing a short demo, and please just bear with me. This is a video, and 1 second, please. Let me just play it. So now we are the test environment of CSS, and you can see NovoMagic on the sidebar. Here's a short introduction about the system for you, and you can click here to navigate to NovoMagic. And this is NovoMagic. Let's first go through each module to see what they are about. So the projects that can be run on NovoMagic are displayed here. The applicable project types include human and mouse mRNA-seq and eukaryotic mRNA-seq with reference. You could also see some analysis from the toolkit. There are 6 modules on the sidebars, which are homepage, project list, task list, user guide video, help, and toolkit. You could either start from the homepage, select the project, or you can click on the project list to see the full list. After clicking on the Start Analysis, the sidebar on the left page has changed to different analysis points, such as rename samples, gene filtering, DEG analysis, gene function analysis, and so on. We could use the DEG Venn diagram as example to run a task. So for each analysis point, there will be user guide video and result interpretation video. After specifying the groups and parameters, we could click on Run. The task can then be checked from the task list. So task list contains all the task history, and you can check the status and analysis result. You could click on a specific task to view the result. Also, it's possible to reset the figure title, color, and a lot more details of the figure. On the other hand, besides analysis points for specific projects, the toolkit is also quite useful, but they are just independent tools and you need to prepare the exact data format to run this tool. But don't you worry, there will be examples for the right data format as well on it. And you could also have a look at the help function, where you can find the quick start menu, which is in a PDF format. You can also submit your issues here. And we also have user guide video, which contains both user guide and result interpretation videos. That's it, and I hope you enjoy this brief demo of NovoMagic. Okay, so now we are the test environment. Sorry. Okay, so that was like a brief demo, and I was just showing one of the functionality, and actually there's still a lot more to be explored. And I guess that there might be still a lot of questions that you're wondering about the system. And here I'm just listing some of the frequently asked questions about NovoMagic. So the first one could be, what is the expression value used for NovoMagic? And just as mentioned, for NovoMagic, it uses DE-seq2 for the DEG analysis. Therefore, we use for the FPKM, whereas for EdgeR is also alternative. And if you have a different request for using different expression value, then that can be done by the bioinformatician from our side as well. Okay. And if you ask about how long will your projects or task or data be available on NovoMagic, and the answer is there's no limitation. So if you have activated NovoMagic, your project and your task, your data will be always available on it. Okay, so you don't have to worry about this part. And it happens sometimes when your task fail and you might be wondering what can you do. And I'm just listing about several selections of the troubleshootings. So for first, like I mentioned on the demo, there's actually a user guide, a user manual PDF about it. And also we have the feedback function where you can just issue all of your questions there, and there will be, we will contact you after we see the issues that you submitted. Also, you can always contact the person you know at Novogene, no matter it is just an email address or it's our account managers or the technical support, and I think they will be more than happy to help you out. Okay, so that's it for my presentation, and I'm happy to take any of your questions.
Thank you, Yue, for excellent presentation. We have definitely had some questions come in. So I'm going to start here with one from Irina asking about what about RNA transcriptomes without reference on NovoMagic?
Okay. This is actually a very interesting question because didn't really touch much on this part. And I think for this, we do have two solutions here. And the first one is if you do not have a reference genome ready, you can first perform a genome assembly using, let's say, the DNA approach, and then the RNA-seq after that would be just the standard one, and you can definitely use NovoMagic on it. But if you do not want to use the DNA approach, but only the RNA approach, then this will be a de novo transcriptome analysis project. And for this is so far not applicable of NovoMagic, but we can definitely perform the analysis from the bioinformatician in our team. Yeah, thank you, Eva.
Thank you. Next question is from Tom saying, can I specify a different software to be used for analysis?
Okay. Okay, so since this question didn't specify on NovoMagic or not, I'll just answer both. So if it is just, using different softwares from our bioinformatician side, actually it is a very, we're very careful about this. So you can just tell us about the software that you're interested to use, and we can definitely help you to test it and tell you about the feasibility and also to adjust the parameters, etc. Whereas for NovoMagic, as you can see, it is now within the scope of just using DE-seq2 and HR for the DEG analysis, whereas for others, so far NovoMagic cannot really do it. But now, as I'm telling everyone here, is that we're still updating the system, no matter from the diversity of RNA-seq analysis that we can provide, also to the scope of different services types. And once you activate the system, those updates will be available for you. Okay. Thank you, Eva.
Lovely, thank you. Next question is from Xinjiang asking, should we remove the duplicates from RNA sequencing, or is this only for UMI tags?
Okay. So the duplicates issue here in RNA is a tricky one. So because normally we do not do the duplicate removal for most of the RNA-seq analysis, because it is a very common issue when you have pair-end reads or the transcripts are shorter, or if you just have a more express transcripts in your data. So just simply removing the duplicates can trigger some real harm to the data. And what we do is we just keep the duplicates. And since you were mentioning about the UMI tag, I think for UMI tag techniques, you could just remove the duplicates as suggested. Whereas for the majority of the RNA-seq, we do not consider duplicates a problem. Yeah, thank you, Aoife.
Thank you. Next question is from Carolina asking, what's the difference between EDGE-R and DE-seq2?
Yeah, so these two tools are very famous one, and the situations we use them are different. So when you have replicates, you would go for DE-seq2, and when you do not have replicates for your analysis, you would go for EDGE-R. Yeah, and that's the differences between DE-seq2 and EDGE-R. Thank you, Aoife.
Thank you. Next question asking, would you advise RNA-seq results confirmation by doing a classical RT-qPCR on a few identified DEGs?
Yeah, so the situation here is that for RNA-seq is giving a very diverse result, and if you could narrow down your analysis to, let's say, a few list of genes, I think to do a real-time qPCR can definitely increase the power of your story, and a lot of the reviewers or journals, they definitely prefer having the confirmation of real-time PCR, but definitely remember to use a very well-designed premise for it. Yeah, so I think we would normally recommend to doing this. Yeah. Thank you, Aoife.
Thank you. Next question is from Ina asking, can I also perform the characterization analysis of RNA-seq on NovoMagic?
Yeah, I think it's so far still a pity that it's a no so far, since I mentioned that the resources that and also the storage of the characterization analysis kind of limits the availability of it, since we will need to start the analysis from the BAM file, which is much bigger after the mapping. And yeah, so far it's a no, but I would say it's definitely included in our standard analysis of RNA-seq. So that's still within the scope of the analysis we provide. Yeah, thank you, Aoife.
Okay, thanks. Next question is from Jiwan J asking, how do you deal with reads that could be assigned to multiple features, like multiple transcripts?
Yeah, so for this question, actually, as well as just showing the status for the summary of mapping, there were reads uniquely mapped, and also there were reads were mapped multiply. And actually, we only take the uniquely mapped reads onto towards the downstream analysis. So we only take the uniquely mapped reads out of it. So that's for the analysis. Yeah, thank you.
Okay, thank you. Next question is from Ella Mofi asking, my project was done in 2020. Am I eligible to use NovoMagic to reanalyze the results?
Yeah, so let me just go back to a bit of the previous slides. Yeah, so far the requirements for using the platform is a bit rigid. So it's requiring for the samples of data arrived at Novogene after the 20th of June of 2020. But if you have done any of your projects after this time, you can still upload the data from your previous projects. So it means that for a lot of the functionality of this platform is not limited to the projects. So you can always upload any format of your data, and you can play around with these functionalities. And if you're, let's say, if some of you might having trouble to, to find the right format for the different analysis, or if you want to confirm with us about whether you're eligible of using it, you can definitely leave a message, and our project coordinator will definitely contact you about the details and tell you the situation about it and try to find a solution for it. Yeah, thank you, Eva.
Thank you. Next question is from Alfonso, asking, how about if I need support from experts, if a reviewer asks for specific analyses or changes in the parameters or software? Do you offer this kind of help?
Okay, so actually, I would take this question into two steps. So for example, we do offer consultation in a way. So if you have any questions regarding how to use the system and what is the best parameters for your study, and you can also collect the suggestions you have from the reviewers, will definitely help you and to give you some advice about how to adjust this. And on the other hand, if you have any other requests about using different softwares, or you want to have a more detailed, or let's say more advanced analysis from the suggestions you have from your reviewers, we definitely have our bioinformatician ready to help you out. Yep. Thank you, Eva.
Thank you. So I think this question will be the last one from Danielle. Can NovoMagic be related to small non-coding RNA analysis projects?
Yeah, so far it's a no. It's a pity though. So for this part, NovoMagic is only available for the messenger RNA sequencing analysis projects. And if you have only performed the small RNA analysis with us, and I think maybe you could have a look at the guest mode of the recon to FPKM TPM and Volcano plot and heat map at this time, and we will definitely give you the best offer. I assure you to make your next analysis for RNA-seq easier and ready for you. And so far, for other kinds of analysis besides RNA-seq, is a no, but we'll definitely keep updating the functionality of NovoMagic. Yeah. Thank you, Eva.
Thank you, Yue, and thank you to everybody else for your time today as well. We hope you enjoyed the webinar and found it useful. Thank you for all the questions. As we mentioned, if we haven't managed to answer yours live, we'll get back to you by email within the next week. In the meantime, we'll email you a link to the webinar recording and the slide deck, along with a short survey that would appreciate you taking the time to complete. The survey will help us ensure that we make future webinars more relevant to you and your research goals. There will also be a link in the follow-up email to a certificate of attendance if you need one. In the meantime, please stay in touch. If you have any upcoming NGS projects, now would be a great time to contact us. Our 30% discount promotion on a large range of our services is coming to an end soon and is now only valid for prepayment before the 15th of December. But if you think you might have samples to send us in the new year, get in touch and see if you can get the discount before you send the samples. So you can also follow us along on Twitter and LinkedIn, or get in touch via our website at novogene.com. And we look forward to seeing you all on the next webinar. Thank you very much.